Hi everyone. Now I'm going to talk about plasmid. What is a plasmid? Plasmid is occurred naturally in bacteria. Inside a bacteria cytoplasm that has DNA lies freely without nucleus. Other than this main genome or main DNA, they also contain a small circular DNA. There might be a few of them, 8 to 10 of them. This small circular DNA we call it as a plasmid. Now you need to know why it is useful to serve as to be used as vector in genetic engineering or in gene transfer. And as a transporter, so for wherever you have a genes, you can, if you want to transfer or transform into the bacteria, for them to help to transcribe and translate to make express the protein out so that you can purify and extract the protein for maybe for patients management or for disease treatment. So you can use plasmid in this case. What is the criteria and why it can be used as a vector? First point, it is acid naturally in bacteria. So they will not have any DNA rejections issues. Wherever you clone the genes into this plasmid, the bacteria will happily accept it into their cells. So there are no rejections since it is acid naturally in bacteria. Number two, it can be transferred easily between species. They have no problem transfer this plasmid within their own species, within their own populations, but the plasmid also can easily transfer to other species. So this plasmid, if the surrounding the bacteria die, other species of bacteria can just take the plasmid easily into their cells. So this makes the transfer of the genes more easy. If you have a vector, maybe from Salmonella bacteria, you modify it, you put it the genes of interest, and now you want to transfer it into E. coli, they have no problem. So this is some of the features that help them to be to have a very why they have a frequently used in gene technology. Number three, it is very small. So it is a small DNA. Compared to large DNA, small DNA is more easy to transfer. And we also can call it as they have a low molecular mass or low molecular weight. So it will be more readily taken out by the bacteria. It is more easier to transform the plasmid into the bacteria during the genetic engineering process. Number four, it is circular. You can see inside here it is round in shapes, meaning that they don't have the beginning, they don't have an ending. It is always in a circle. And circular DNA is more stable compared to linear DNA. They are not easy to degrade. And when you insert the genes inside and you want to store it for a long time, it can store for many years without any DNA degradations problem. Number five, it is double-stranded DNA. Double-stranded means they have two strands, and this is the same characteristic with all your prokaryotic, eukaryotic DNA or genes. So it enables you to clone your desired genes into complementary to this plasmid since it has the same characteristic. So there are no problem in inserting the genes or no problem in creating the complementary end or the sticky end using the restrictions enzyme. Number 6. One of the main features why plasmid is frequently used as vector is because plasmid can replicate independently. Independently independent of the DNA here. So they are not depends on the main genome DNA replication. So whenever the cells want to replicate, this DNA inside here is going to have binary fissions. So they will going to copy and they will produce two of these identical DNA and separate into two daughter cells. At the same time, plasmid also will replicate by their own. So they replicate independently, and in genetic engineering, we put our gene of interest inside the plasmid that will not really affect the replication's ability of the whole bacteria. So if you put inside the genome, maybe some of the phenotype of the bacteria will change. But since they are replicate independently, it will not have a huge impact on the bacteria. So they can still growing as normal, but while they are transcribing and translating the genes that you have been inserted inside the plasmid. Last point, why it is used as vector, because they contain special genes that contains antibiotic resistant genes. So these antibiotic resistant genes can act as a gene marker. As marker for a researcher during the genetic engineering, if they want to find out whether the bacteria has successfully taken in the plasmid, they can use this antibiotic resistancy to check whether differentiate which bacteria have taken out plasmid or which one does not. Now let's zoom in into the plasmid genes. This is the bacteria, and inside it you have plasmid. If you zoom in into this plasmid, you know it is a double-stranded, means they have one strand that is circle in shape because it is circular, and you have another strand here to form a double-stranded DNA of the plasmid. For plasmid to be a successful vector, they must have the below characteristic. If some of the plasmid they might lack of this characteristic, the scientist or the researcher can just edit it artificial in lab. They can either insert some of the promoter, insert some of the multiple cloning site inside to help or ease in their gene transfer. Now the first feature is they must have a segment of the DNA sequences that contain origin of replications. So this is the site where the replications occur, is where the replications, the locations where the replications start. Because this is a circular DNA, they have no starting point or no replication starting point, so they must have these origins of replications only exist in plasmid. And this allows this plasmid to replicate independently in the bacteria. Other than these origins of replication site in these DNA sequences, now this is double helix, this is a double-stranded DNA. So you have, like say this is A, then this must be T. If this is C, this must be G. So this is a double-stranded DNA. Now a segment of these DNA sequences must contain promoter. If they don't have the promoter in front of the genes, there are no transcription and translation happen. So the promoter is the site where the RNA polymerase go and bind and initiate transcriptions. Right after the promoter is always your gene of interest because you need promoter that help you to have transcriptions of your genes. Now in this case, for the genes to insert into plasmid, you need another segment of DNA that is called for multiple cloning site. They have a specific DNA sequences for this one, and you have learned a lot about the restrictions enzyme. They cut at a specific site. So let's say you have this AACGTT, that is the restriction site, for example, for BAM-H1 enzyme maybe. So you will have this DNA sequence is inserted into this site. So this is one of the restriction site, and you might have another restrictions enzyme, restriction site here for another types of restrictions enzyme, another restriction site here. So you have multiple cloning site here, multiple restriction site, so that your restriction enzyme can cut and you can clone your genes into these regions. Now you need to have multiple restrictions enzyme site because they can give options for scientists to choose which one is the preferable restrictions enzyme to digest and to clone, and which locations is desirable to clone the genes. So you have multiple choice inside here. Other than origins of replications, promoter, multiple cloning site, another important features that a plasmid might has is that might has a marker genes in their DNA segment. Now the marker genes are the ones that allow differentiations of cells that have the plasmid with those that do not have this plasmid. Now marker genes could have a lot. Either is antibiotic resistant genes, or could be example like GFP protein, the fluorescent genes, or the GUS protein, the GUS genes that were called for an enzyme. They will give them color. So all these markers are the ones that allow identifications of cells that have taken up the plasmid. Let's say you have a flask here. Inside this flask, you have put in a plasmid that you have, let's say this is the plasmid, and you already cloned the genes of interest inside. You have another plasmid here, or recombinant plasmid with the gene of interest. Another thing is inside these solutions, you have bacteria. If you mix these two together and you follow like a heat shock method or calcium chloride method, some of the bacteria will take out the plasmid, some are not. Not all of them will take out. So they will have two results. One of the bacteria will look like this, without any plasmid inserted inside or without any transformations of the plasmid. And another plasmid might have plasmid with the gene of interest. So this is the gene of interest, this is the plasmid. This is what we want, this is the purpose. For example, if you want to have modified these bacteria so that they can produce human insulin protein. So you put the insulin genes into this plasmid and transform into the bacteria. And this is the successfully transformations of the bacteria. Let's say inside this plasmid, this is the gene of interest. You will have put in another site here that is the antibiotic resistant gene. For example, here you have ampicillin resistant gene. And now, while you are transforming the bacteria, you make these solutions containing ampicillin antibiotic. So they will help you to select which bacteria successfully contain the plasmid with your desired genes or which one do not have. So for example here, they do not have plasmid. These bacteria do not have plasmid, so they also do not have ampicillin resistant genes. So can they survive in ampicillin solutions? Cannot, so cannot survive. Since these solutions containing ampicillin. And this type of bacteria that have successfully transformed the plasmid, that have the ampicillin resistant genes, they can survive. So we say marker gene is the one that help you to differentiate which bacteria has taken out the plasmid, which one is not. So this is the one that not successful transform. This is which cells, this is the cells that have taken out plasmid. And how you can decide whether this is successfully transformed or not transformed, because you will see those any bacteria that is survive in these solutions. This is the bacteria that already transformed with the plasmid. So they also contain the gene of interest, and this bacteria you can then transfer to another flask and go for mass productions. So this is the functions of the marker genes.